Okay, take another couple seconds to pick some answers for this, and then we'll go over these questions. Okay. Okay, so we were thinking here about our graded potentials. And can everybody see the results now from this poll? I think so. Yeah, okay. So when we were talking on Wednesday, we were talking about graded potentials and how what's happening in our neuron is that we're going to have a stimulus coming in from another cell. Often that'll be a neurotransmitter. And what that graded potential really is a change in the charge of the neuron. So a change in that membrane potential. So our resting potential for a neuron, we remember, is -70. So we're going to see a slight change in that membrane potential, either becoming more positive if it's an excitatory stimulus, or more negative if it's an inhibitory stimulus. So our graded potentials don't change anything about the equilibrium potentials. The equilibrium potentials, we remember, are just like telling us about the forces on sodium and potassium. So we have an equilibrium potential for sodium that's at +60, telling us that sodium wants our membrane potential to become positive. So sodium is going to move until the inside of the cell hits +60 if we let it. And the equilibrium potential for potassium is negative, like -94-ish. So potassium likewise will move to try to get the neuron to -94. But when we change the permeability of that cell, when we allow sodium and potassium to flow, they're going to flow in the direction that we can figure out from our equilibrium potential, but it doesn't change the equilibrium potential itself. What we're changing is the membrane potential. So whenever we measure like the charge inside the cell versus the charge outside the cell, what we're measuring is the membrane potential if we're measuring something in real time. Okay. So the way we manage to change the membrane potential in response to these stimuli is that we're going to change what can flow in, what can flow out, how fast it can flow in, how fast it can flow out, which means that we're changing the membrane permeability to sodium and potassium. Sodium, if we're talking about an excitatory stimulus, or potassium if we're talking about an inhibitory stimulus. So the reason this works in order to change the membrane potential is that at rest, we have, right, our neuron. The membrane is pretty highly permeable to potassium, not very permeable to sodium. The total membrane permeability to either of those ions is dependent on the leak channels that are present. There are like 25 times more leak channels for potassium than for sodium. So if we have a stimulus that opens more sodium channels, we know that sodium wants to flow into the cell. It wants to make that cell positive. So when we open more sodium channels, that cell becomes more positive, which we call an excitatory stimulus. That direction we're calling excitatory. And we'll also see that when we talk about the change in charge, so the change in the membrane potential, we'll refer to this action of sodium entering the cell as depolarization. So depolarization is moving us away from our membrane potential. At rest of -70, we're becoming more positive. So as an excitatory stimulus opens channels allowing sodium to enter, we're depolarizing the cell. So the opposite is an inhibitory stimulus. So an inhibitory stimulus is still going to change membrane permeability, but what it's going to do instead is open potassium channels. Potassium wants to leave the cell, but potassium is positively charged. So as potassium leaves the cell, it's taking charge out, so it makes our cell more negative. So we call this hyperpolarization. We become even more polarized than at rest. And this is basically going to make it harder for us to reach the threshold to fire off an action potential, which is why we refer to this as an inhibitory stimulus, because it's making our job harder for signaling. We're really trying to stop a signal in this case. Does anybody have questions about these before we move forward? Okay. So when we left off, we had talked about graded potentials and we'd drawn out temporal and spatial summation. So in addition to understanding what's happening with the graded potential, like the fact that our membrane potential is changing, we want to make sure we remember that we can add up graded potentials, that they decrease as we move farther away from the location of the stimulus, either in time or in distance. But if we have two stimuli coming together, either one after each other in quick succession or hitting the cell at the same time but in different places, we're going to be able to add together those stimuli. That's because each stimulus is opening channels. So every time we have a stimulus, we're going to affect some amount of change to that membrane potential. Okay. So now that we're moving forward and starting to talk about action potentials as opposed to graded potentials, we're going to want to think about some different kinds of ion channels. So when we left off, we were looking at the slide of different types of ion channels. I just want to point out to you as a quick reminder where we've seen these channels so far. So when we were talking about the resting membrane potential, we were really talking about leak channels. So when we talked about setting it up by allowing potassium to leave the cell, a little bit of sodium to enter the cell, and then maintaining that resting membrane potential by basically having an equilibrium between the sodium flow and the potassium flow. All those channels that we were looking at were leak channels. So those are going to be present all the time, everywhere, and they're always open. What we were talking about really when we were talking about graded potentials, so we're talking about stimuli coming in and changing the membrane potential in response to, for example, a neurotransmitter. What we're mostly talking about there, or what we want to associate that with, is ligand-gated channels. So ligand-gated channel is not always open. It can be open or closed. The way we open it is we have some other molecule bind onto it. So in the example of graded potentials, we might think about a neurotransmitter binding to this ligand-gated channel. It opens and changes our membrane potential a little bit, producing one of those graded potentials. So correspondingly, the place that we're going to see those ligand-gated channels are the part of the cell that receives signals, which are the dendrites in the cell body. So what we're moving into now are voltage-gated channels, and they're a little more complex, and we're going to look at them in detail, and specifically we're going to spend some time looking at the sodium voltage-gated channels, because those are the ones that are better understood at this point in time. What we want to keep in mind is that our voltage-gated channels, right, we see that word voltage there. This means that they're going to open and close in response to electrical charges, by which we really mean is that they're going to be opening and closing in response to changes in the membrane potential. So when I've mentioned that we have a threshold that we're trying to get up to, this is why. We're trying to get up to the threshold that's going to trigger these voltage-gated channels. So we'll see that the voltage-gated channels are more prevalent in the places that are associated with sending off our action potentials, which are going to be the axon, and especially the axon hillock, which is just the beginning of the axon. And finally, when we start talking about synapses, we'll come back to calcium channels, but we're not going to focus on those quite yet. Okay. So when we're talking about changes in the membrane potential, we're always talking about changes in what ions are flowing. Like that's how we change the membrane potential. That's how we produce voltage in the cell. That's how we track charges. So we're always, always, always secretly talking about membrane permeability to ions, even if what we have a picture of in front of us is a picture of the membrane potential, a picture of the charge. Okay, which means that we have to know what channels are allowing ions to flow, because whatever ions are flowing is going to determine which direction that membrane potential is going to shift. I do want to say that as we're tracking these channels, fortunately for the action potential, we actually don't have to sort of rethink what direction each ion wants to go. Now that we've set up our membrane potential, now that we've set up the equilibrium potentials for sodium and potassium, we can kind of shorthand remember sodium wants to go into the cell, potassium wants to go out. That's going to be true basically in all cases as we track the channels for our neuron. So we hopefully won't have to do math again about the EDF, CDF, or ECDF, although we should still remember it because we've spent a fair amount of time on it in this unit. But moving forward, potassium wants to go out, sodium wants to go in. Okay, so potassium has two types of channels that will allow potassium to go out. The first one are the leak channels that we've talked about for setting up the resting membrane potential. They're always open. And the second are going to be the voltage-gated potassium channels, which are going to be triggered by changes in the membrane potential, so triggered by voltage. So what we want to remember about those voltage-gated channels, because I said that's really what we're thinking about now, is that our voltage-gated potassium channels are closed at the resting potential. So before we start out our action potential, those voltage-gated channels are closed. The same thing is true for our sodium channels. The voltage-gated sodium channels are going to be closed at rest. And the membrane is not very permeable to sodium at rest, so we can kind of wave away the leak channels for sodium. So we really just want to think about the voltage-gated channels when we're thinking about the area of the axon specifically. Okay. So we're going to take a look at what a voltage-gated sodium channel looks like. So we can kind of think of a voltage-gated sodium channel as having two doors. Okay, so we have our protein. All channels are proteins in the membrane. Remember, they're transmembrane proteins. It's got two doors. We got one, I'm gonna pull out my annotation tool, or just the pointer, I guess. No, oh, my marker. Okay, so we have our first door here in the center of our channel. We're going to call that the activation gate. And then we have a second door on the inside of the cell. Okay. We've got a second door here on the inside of the cell on the intracellular side called our inactivation gate. So what we want to know is that when we start out at the resting membrane potential, so before we've changed anything, our activation gate is closed and our inactivation gate is open. So this activation gate in the center is what we're trying to trigger to start out. So our inactivation gate is already open at the beginning. Our activation gate is blocking sodium from entering the cell. So we got to open that activation gate in order to start our action potential. Okay. So the way that we can open that activation gate is by reaching this threshold of -55 millivolts. Okay. So we do that by having graded potentials, excitatory graded potentials that depolarize our cell. Remember, depolarizing means the cell gets more positive. -55 is more positive than -70. So if the membrane depolarizes to -55 in response to our graded potentials, this activation gate is going to open. So negative 55 is our threshold. And whenever we see a graph of the action potential, if we're charting the voltage, we're going to see that threshold kind of marked on there. Okay. Once this activation gate is open, sodium starts to rush in. Now this is because we're really far away from our equilibrium potential for sodium. Remember that the equilibrium potential for sodium is positive 60. The only reason the cell isn't more positive than it is because that membrane was not very permeable to sodium. So sodium really wants to move. And once we reach negative 55, open this activation gate, we let it, sodium flows in. As we allow sodium to flow in, we're going to start rushing towards the equilibrium potential for sodium. So as this activation gate is open, our membrane potential is going up and up and up, becoming more and more and more positive. It's actually going to get all the way to positive 30 before this channel starts to close. So we're not going to get all the way up to our sodium equilibrium potential. We're not going to hit positive 60 because this channel has basically a timer on it, right? These two doors are timed, they slide back closed at around 30 millivolts. So if we move to the second picture, so this first one on top is our resting membrane potential. It shows our activation gate closed, our inactivation gate open. And I find this phrasing a little confusing, but you can note that it describes the activation gate as closed but capable of opening. The reason it specifies that it can open is that we'll see at the end of our action potential, we have a refractory period where one of these gates actually is unable to open for a period of time. So at the beginning, at our resting membrane potential, this gate is closed, but it can open. When we hit our threshold of negative 55, when we depolarize to negative 55, that activation gate opens. So that's what we see in the second picture here on the right. And so now we can see our inactivation gate was already open. Our activation gate has now opened because we've hit the threshold voltage. So sodium is rushing in. So we see that it's drawn on, our sodium flowing through the two gates. And then we're going to move to our third picture. So after about a millisecond, our inactivation gate closes. Okay, so now we're not talking about that top gate in the middle of the protein. We're talking about this bottom one on the intracellular side. So you can see this kind of like tail curling back up to close up this channel. That's our inactivation gate. And now that it's closed, sodium can't get into the cell anymore. Note the activation gate is still open here at the end. The only thing closed is the inactivation gate. And it's actually incapable of opening for a little while. So it's closed, can't open until we get back to the resting state. A thing that I do want you to know also about the inactivation gate is that this shows us these three pictures in a nice sort of wheel. So step one, step two, then back to this first step. But actually the action of both of these gates is a response to the depolarization. It's basically that the action of the inactivation gate, so this bottom one on the intracellular side, is on a delay. So it's also responding to the voltage change, but it's just slower. Okay. So questions so far about these gates, the activation gate, the inactivation gate, and how they're affecting the movement of sodium. Okay. So I want you to note, as we're moving on to the next one, that when we have these voltage-gated channels open, if we were tracking the permeability of the neuron to sodium, which we will do, so the permeability of that membrane, when we open these channels, when we have more channels open, So at this sort of second timestamp, our permeability of the membrane to sodium is going to be higher. So we've increased the permeability of this membrane to sodium by opening this gate. So we're going to see a picture of that. So I just want us to sort of keep in mind this picture and associate it when we see that increase in the permeability to sodium. Okay. So this is a little kind of summary about the activation gate and the inactivation gate in words instead of pictures. So the activation gate is triggered by this change in voltage. It opens at the threshold and it keeps opening. So like neighboring voltage gated channels open during depolarization. So once one channel opens because the membrane potential near that channel has hit threshold, that channel opens, allows sodium in, which means that we've gotten our two neighboring channels up to their thresholds. So they're going to open. So we'll actually talk about the opening of these voltage gated sodium channels as being regenerative, or describing them as positive feedback. Okay. And that's because as we allow sodium in, we become more positive. So we're depolarizing more and more areas of the membrane, which means that we're triggering more and more voltage gated sodium channels to open. So our inactivation gate, the other one, the one on the bottom, is slower. So it's responding to voltage, that change in depolarization, that threshold, but it's also responding to time. So it hears that signal. It knows that we hit -55, but it's just kind of slow. It doesn't catch up for a while. So it doesn't close until about a millisecond later. And in that millisecond, our sodium concentration inside the cell has rushed up. The membrane permeability for sodium has rushed up. Our membrane potential has rushed up. And that's why we get to +30 before the change to our membrane potential goes back down. So part of the reason that we're going to see the membrane potential not get any higher than +30 is because that's the point about when these inactivation gates start closing, which means that sodium is going to stop getting in, which means our sodium permeability is going to go down. Okay. So the voltage and time both affect the action of this inactivation gate. And when we see the phases of our action potential, we'll see that it's both opening and then also closing during depolarization. So during this depolarization phase, we have some time points where it's open and then some time points where it's closed. Okay. So I think your book doesn't have this picture and doesn't in general have a picture of a voltage gated potassium channel. This is because just less is known about them generally. But the way we think they work is a little simpler than the sodium channels. So they still respond to voltage, but the way they respond to voltage is that they basically just have one gate. So at about the time point of the time that the sodium inactivation gates are closing, sorry, going back here. So we saw that these sodium inactivation gates were triggered after about a millisecond around the time that the voltage gated, or sorry, around the time that the membrane potential gets up to +30. That's the same time we're going to see these potassium channels opening. Okay. So the potassium channels are opening at the same time that that inactivation gate for the sodium channels is closing, which means that if we're looking at sodium permeability, so sodium permeability was going up because we were opening those activation gates. And then the permeability is going to flatten out, potentially start to drop as we close the inactivation gate. But potassium is going to be different. So potassium permeability is higher than sodium at the start, right? At the resting membrane potential. Nothing happens to it during that first part. So as sodium is rushing in, nothing's happening with our voltage-gated potassium channels, but at about the same time as the sodium inactivation gates close, the potassium channels open. And at that point, our membrane permeability for potassium goes up. Potassium wants to get out of the cell. We remember that our potassium equilibrium potential is highly negative. So potassium is going to rush out of the cell following its electrochemical gradient. This means that we no longer have sodium coming into the cell, and now we have a lot of potassium going out. So the membrane potential of the cell is going to go back towards its equilibrium potential. So we're going to call that the repolarization phase, because we're going back. And the way we manage to go back is we're having these potassium channels opening. Okay. So this is specifically talking here about the voltage-gated potassium channels, these 3 bullet points, saying that those potassium channels that we're looking at, we think they just have one gate. Like the sodium inactivation gate, we think that they depend on both voltage and time. So we think that the way that that gate for the potassium channels is working is that it's still responding to that initial depolarization, but like the inactivation gate, it's just kind of slow. And this is a form of negative feedback. So our positive feedback was this sort of runaway movement of sodium. So the depolarization to -55, which is our threshold, and then letting sodium in, which means that we're depolarizing more and more and more and more and more. And whenever we can describe something as more and more and more and more, we think of that as positive feedback. Negative feedback here, we're turning off this change in the membrane potential. We're getting ourselves back to rest, back to the resting membrane potential. So this is negative feedback, bringing us back to that initial sort of steady state. So here we can see that this channel is closed at the resting membrane potential. It's open after depolarization, eventually in order allowing us to repolarize. And then eventually it's going to close back up during the refractory period. Okay. So if we were to chart out the permeability and the membrane potential, it would look something like this. So I'm going to read this chart to you first. So here on the left, it says membrane potential. And here on the x-axis, we have some timestamps. In red, we have the action potential. So the action potential is the change in the membrane potential over time as we're sending off the signal. So we can see that we started out at our resting membrane potential of -70. Somehow we got up to -55. We added up some graded potentials. This triggers this positive feedback where we're opening all these voltage-gated sodium channels. Sodium is rushing in, that's opening more and more voltage-gated sodium channels, opening, opening, opening. We then have those sodium channels start to close because the inactivation gate that's on this delay starts to swing closed. And at the same-ish time, we start to trigger our potassium channels. The voltage-gated potassium channels open, but potassium wants to go the other way than sodium. Potassium wants to go out of the cell. So this sort of combo hit drops the membrane potential down, 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 down. We're actually going to overshoot our resting membrane potential at the end. We're going to have this period of hyperpolarization after this spike of the action potential before we get back to our resting membrane potential. Okay. So this red line is just tracking the charges. And then these two bluish lines are tracking the membrane permeability. So we can see we have like a second y-axis. So the y-axis for these permeabilities is on the right-hand side. So that's why it's abbreviated here, P in parentheses. So we'll start with this kind of darker blue, maybe even purple line. So this is showing us the permeability of sodium. So here, and we're going to compare it here to the permeability of potassium, which is this kind of lighter blue line. So we can see that at the resting membrane potential, so down here at time point A, we can see that the membrane is more permeable to potassium than to sodium. So remember that that's part of why we set up the membrane potential the way we did. That's part of why the membrane potential is negative, because this membrane is permeable to potassium more than sodium. As we go up to negative 55 using graded potentials, right? Our graded membrane potentials were in response to stimuli, excitatory stimuli, get us up to negative 55. So these excitatory stimuli allow some type of sodium channels to open, probably ligand-gated sodium channels. Once we get to negative 55, this opens the activation gate on our voltage-gated sodium channels. So as the activation gate on the voltage-gated sodium channels open, we can now consider the membrane more permeable to sodium, right? Because all those channels are now opening. So this permeability to sodium is shooting up because when one voltage-gated channel opens, it makes the membrane potential a little more positive, which means it's going to trigger neighboring voltage-gated sodium channels. So go up and up and up and up. Here we can see at time point C, what we have happening is that our permeability for sodium is going down. And the reason for that is that our inactivation gates on those voltage-gated sodium channels is closing. So the activation gate is still open, but we have two doors. And in order to actually get inside, you have to go through both doors. So once that inactivation gate starts to close, the permeability of sodium goes down. And not every inactivation gate closes immediately all at the same time, which is why we have a slope down instead of just a straight line. We can see at around that same time point, so still at this time point C, we start to see the permeability to potassium going up. That's because we have those voltage-gated potassium channels starting to open around the same time. So they were also kind of on a time delay from this threshold depolarization. So our sodium permeability is going down, which means that less sodium is coming into the cell. Our potassium permeability is going up, which means we have potassium rushing out of the cell, which is why we have this overall action potential, the total sum of the difference in charges across the membrane going down. So we have fewer and fewer positive charges inside the cell because we don't have sodium coming in anymore, but we do have potassium leaving. And we can see here that it takes a little while for those potassium channels to close, right? So our membrane permeability to potassium is elevated. We got down to negative 70, but oops, now we have to actually shut the potassium channels. So we have this slight area of hyperpolarization after action potential because we're shutting down those channels. And then eventually we go back to normal, back to our original state. Questions about this picture? We'll come back to it a couple more times, but this is really a summary of almost everything that's happening as we're talking about the action potential. So this is kind of really crucial. And if you can understand what's happening here, you should be really solid on understanding why this action potential is happening and how we're kind of sending this spike in charge. Okay. So we're gonna run through this in some words now. So what we're tracking when we're tracking our action potentials is membrane changes. So membrane potential changes. So we say a membrane is excitable, an excitable membrane, when it has the ability to generate an action potential. So if we can do this, if we can raise our membrane potential, spike it up positive, that membrane's excitable. An action potential is specifically a rapid and large depolarization. So that spike in charge, that's our action potential. And it's used for communication, and specifically for kind of longish distance communication. So an action potential can travel quite far. We'll see nerves that go, all the way from your brain to your stomach. So an action potential can be used for long distance communication by sending this large wave of depolarization. So in a neuron, specifically if we're thinking about where this action potential is happening, we're talking about that long skinny axon. So the action potential is traveling along the axon. It starts when we have graded potentials, changing the membrane potential in the cell body, the soma. Basically, it's going to start at the axon hillock, which is the beginning of the axon. So the little kind of hill, the little pyramid at the beginning of the axon, that's where it's going to start. That's where we'll first see this rapid, large depolarization. And then it'll travel in a wave down the axon. And eventually it'll get to the axon terminal, the termination of the axon, just the end of the axon, which is where we'll see it releasing neurotransmitters and doing its communication action. So we'll get into that when we start to talk about synapses. So when we begin our action potential, and this is just looking at that same thing I talked about with that blue and red graph, but in more words and slightly different pictures, but it's all the same concept. So the action potential starts when we reach this threshold membrane potential. So when we hit negative 55 millivolts, that's our threshold depolarization. Opens up our voltage gated sodium channels. So specifically, we're opening those activation gates. Sodium rushes in. Okay, so we can see this on the top in this picture. They've separated the diffusion of sodium and potassium from our action potential. So what we see on this top image is the change in the membrane potential, so our action potential. And what we see on the bottom image is the diffusion of sodium and potassium. So on this top image, we can see that we have this spike in charge, and we can see that that's corresponding to number 1 on the bottom, our gated sodium channels opening. Okay. So this is a positive feedback loop. It causes more and more sodium to come in. It causes the membrane potential to shoot up. Now this overshoot, I'm not sure if the word overshoot is strictly necessary there. It gets over negative 70. That's not surprising. Gets all the way up to positive 30. And we call this phase of the action potential depolarization. So the first part, and we'll see that picture with the areas of the action potential kind of highlighted in different colors. But the first part is depolarization. As we continue, so once we hit this positive 30, our 2 gates that were kind of on a lag start to respond. So the one that's on a lag on the sodium channels is that inactivation gate, so it's closing. So here we see 2A on the bottom. The inactivation of sodium channels begins. Okay. Inactivation of that sodium channel is beginning, which means we don't have sodium coming in anymore. The other thing that we have happening here, 2B, is our gated potassium channels are opening. Remember, potassium wants to flow out of the cell, not into it. So it's flowing in the opposite direction than sodium. So it's going to pull the membrane potential down. So we have this membrane potential going down. We're going to refer to this as repolarization. So this is the part of our action potential that's a negative feedback loop as we start to pull that charge back down to the resting membrane potential. Can also look at this in table form. So this is actually also things we've talked about. I've pulled that little graph in on the bottom here that we were looking at. But this is showing us are phases of the action potential. So it's named them here. So we start out at rest. So we start out at our resting membrane potential. Then we depolarize. So depolarization happens after we hit the threshold of -55. We have a bit of a time delay. The membrane potential gets all the way up to +30. At that point, our gates start to change, so we repolarize, so we come back down. We have a slight period where our potassium channels are, some of them are still open, so we have hyperpolarization happening after the action potential. So sometimes it's specified this way, calling it after hyperpolarization. Okay. So we can see that it's charted out the numbers, right? -70 at rest. Depolarization, this is kind of two parts. So the first part of depolarization is the -70 to -55 as we hit the threshold. And then we have the -55 to +30 as the sodium is rushing in. And then we have 30 going back down to -70. And then we have this slight period where we dip down super close to the equilibrium potential for potassium because we're letting a lot of potassium out. This shows us also the state of our gates for the sodium channel and the potassium channel and the influence on the sodium flow and potassium flow. So if I were you, I'd actually maybe make a blank version of this chart and try to fill it out yourselves as practice for the exam, because this is really a full summation of everything that's happening during these phases. So the voltage-gated sodium channels are closed at rest, and that's because the activation gate is closed, even though the inactivation gate is open, which means that the only sodium flow we have is a little bit going through leak channels that are in the cell body. Our potassium channels are also closed at rest, but we have flow of potassium out through leak channels also. And that's why we're having this balance. That's why we stay at this membrane potential of -70. As we depolarize, we open the inactivation gate, we open the activation gate. The inactivation gate was already open, which means we have a ton of sodium come in. That's why we're shooting this membrane potential up. Our potassium channel is still closed, so it can't make up for it. So the only potassium we have going out is through leak channels, and there's not much of that happening. So that's how we managed to get all the way up to +30. And we have the repolarization happening as the inactivation gate for sodium closes, the voltage-gated potassium channel opens, which means we're stopping the flow of sodium in, and we're increasing the flow of potassium out. This after hyperpolarization is just kind of the effect of the fact that these voltage-gated potassium channels take a little while to close. So you can see here, it doesn't really say open or close. They're in the process of closing, which means we still have a pretty high potassium outflow, but those voltage-gated sodium channels are closed. So we can see that our voltage-gated sodium channels by this point have kind of returned to their resting state. So they're closed because the activation gate is closed. And those are the four phases we want to think about our action potential. So I'm going to do a final poll. I think I have one on the action of these gates, and then we'll stop here. So if we compare them to the chart, we can kind of see the answers to most of these. So our voltage-gated channel that has both an activation gate and an inactivation gate. Here's the sodium one. So our sodium channel has 2 doors. So we have to track both doors because both doors have to be open in order for the sodium to flow through. The time point at which they're both open is depolarization. So for number 2, which voltage gated channel is open during depolarization? It's sodium. Okay. So number 3, what's closing about a millisecond after depolarization? So the thing that closes as we repolarize is the sodium gate, and specifically the sodium inactivation gate. So sodium can't flow in anymore. The thing that opens is the potassium gate, allowing potassium to flow out, also happening during that repolarization phase. So those 2 things, number 3 and number 4, the sodium inactivation gate and the potassium gate, are working together to pull us back down towards our membrane potential, our resting membrane potential. Okay, so that's it for today, and we'll pick up here on Monday. I'll stick around for questions as usual.